Okay, we're moving on to part E. So now we have successfully cloned the gene for SUVAR2, and the sequence indicates that it has a protein domain with sequence similarity to histone methyltransferases, and a second domain that has sequence similarity to Argonaute. What is the significance of these two domains? So let's recall, our suppressor of variegation was a suppressor that caused our mutation of the white gene that was embedded in the heterochromatin, giving us our variegated phenotype. So our mutant was less variegated. That means there was less heterochromatin being formed in our mutant. Therefore, we could infer that the wild type function of SUVAR2 is to make heterochromatin. So when we finally cloned the gene, we could sequence it and found that there were two protein domains associated with the protein. One was a histone methyltransferase, which I write here as HMT, and we know that histone methyltransferases are a subset are required to make heterochromatin. They methylate some of the lysine residues that are on part of the histones. The second domain was Argonaute, and so we know that Argonaute is involved in gene silencing pathways as well. Argonaute binds to small interfering RNAs, and we talked about one such pathway, the RITS, using the RITS complex, where this was an Argonaute that was associated with chromatin remodeling enzymes that would be brought directly to the site of the chromosome where its action would be. And so now this would be the answer, is that the HMTs add methyl groups to histone lysine residues, and they result in repressive chromatin, and the Argonaute domain suggests that the RNAi pathway is involved in gene silencing.